So we're going to look at carbohydrates today in this video. So there are three types of carbohydrates we need to look at. Our monosaccharides, mono meaning one, okay, so the single unit, the monomer. Our disaccharides, which mean two monomers joined together, and polysaccharides, which means many monosaccharides joined. So monosaccharides we're going to look at are glucose, both alpha and beta, galactose and fructose. We won't look at the structure of galactose and fructose, but you need to know they're monosaccharides. Disaccharides are maltose, sucrose and lactose, and our polysaccharides are starch, glycogen and cellulose. So we're going to start with our monosaccharides, and we're going to start with the general formula. The general formula of our monosaccharides is CH2O, all in brackets N, where N can be any number. So for example, if N is 3, that would be called a triose. If N is 5, that will be called a pentose sugar. And there are two that you come across, ribose in RNA and deoxyribose in DNA. And when N is 6, it's called a hexose sugar. And those are the ones we're going to concentrate on. So we're going to concentrate on the CH2ON ones, in other words, C6H12O6, which should be a really familiar formula for you. You should have come across it as glucose in photosynthesis and in respiration at GCSE. So I said to you that we're just going to look at the structure of glucose, although you need to know that both our fructose and our galactose are also monosaccharides, but we're just going to look at glucose. So we're going to look at alpha and at beta and look at the difference between them. So we said there were 6 carbons, and it's a ring structure. So what you need to start off doing is drawing a hexagon, but within your hexagon should be an oxygen as one of the ends. So you'll notice we only have 5 carbons at the moment. Starting labeling from there, 1, 2, 3, 4, 5. Our 6th carbon sticks up here, which is over there. You then have on each carbon H and OH, and I'm going to concentrate on the ones that are over here on carbon 1, because that's the one that we're going to be looking at in detail. So there's my H on the top and my OH on the bottom. And then I'm going to go and draw in the other ones. H, H, OH. That is alpha glucose. Now, what about beta glucose? Well, beta glucose starts off looking much the same. I'm just going to write CH2OH there. The difference is that on carbon number 1, the OH is on the top and the H is on the bottom. And that's what makes the difference between alpha glucose, hydrogen on the top, beta glucose, hydrogen on the bottom. And it's going to be really important when we come and join them together. Now, in terms of our monosaccharides, and indeed your disaccharides, so all of these are all sugars: glucose, galactose, fructose, maltose, sucrose, lactose. They're obviously used in respiration. They're needed for energy release. That's going to be their function. So how do we go from a monosaccharide to a disaccharide? So first thing important to know is what our disaccharides are made of. So now the formula for a disaccharide is slightly different to obviously the monosaccharides. So let's think about what we've done. We've taken 2 C6s, H12s and O6s, and we've joined them together. But what we actually get is C12, H22, O11. Okay. Now, those of you who can do some maths, if you imagine you've got C6H12O6 plus C6H12O6, giving you C12H22O11, chemical formula you must have something left, and the answer is H2O. When you join two monosaccharides together, you'll make your disaccharide and you will make water. So how do we do it? Well, let's start with maltose. And maltose is made up of two alpha glucoses joined together. So let's draw them out and join them up. So I'm going to draw an alpha. I'm going to draw an alpha glucose. I'm not going to draw everything in it because it's just going to make it really complicated. Okay, I'm going to concentrate on the H and OH on the end of carbon number 1 and carbon number 4. I'll draw a second alpha glucose next to it. I'll draw it in a different colour so we can easily distinguish between the two. So we've got our carbon 4 there. I know they're alpha glucoses, so I'm drawing hydrogen on the top. Okay, and let's see what happens. We know we've got to be removing water. The easiest place to get water from is down here. We've got an OH and an OH. So if we take hydroxide from one and hydrogen from the other, it doesn't matter which way round, then you will end up with H2O. And if you then redraw what you've got now drawing absolutely everything in it. You've got that. And what happens is your oxygen and carbon join together. And this bond over here, carbon-oxygen carbon bond, that is called a glycosidic bond. I'll spell that. Glycosidic bond. And more specifically, because it's joining carbon 1 and carbon 4 together, because remember this was 1 and this was 4, it's called a 1,4 glycosidic bond. So a disaccharide is two monosaccharides joined by a glycosidic bond formed when water is removed in a condensation reaction. You will always have a condensation reaction when you join molecules together and create a chemical bond. If we wanted to separate them, you want to go back in that direction, we would need to put in water, because it's obviously in the opposite direction, and you would have to split that water up into the hydroxide and the hydrogen and put them back in. So that way is called a hydrolysis reaction. So condensation joins things together. Hydrolysis breaks things apart. Lyse means to break. So you're using water, hydro, you're breaking up water and breaking it apart. Okay, so if you go back to the chemical reaction here, obviously if you want to go in the other direction, which you can do, then you're going to need water for that reaction. That's your condensation reaction in that direction and your hydrolysis reaction in that direction. So maltose is alpha glucose plus alpha glucose. Sucrose is made up of your glucose, also alpha, and your fructose joined together by a condensation reaction and a glycosidic bond. And lactose is made up of glucose joined to galactose. Again, condensation reaction, glycosidic bond. So before we move on to polysaccharides, I want to think about the Benedict's test. Now, here is a picture of the Benedict's test, which I'm sure you're used to seeing from GCSE. Okay, here is your Benedict's, like so. Make it bigger. Okay, blue Benedict's solution. When you add it to your sugars and you heat it, okay, so you add Benedict's, heat, and it goes from blue to brick red. As long as there's large enough amounts of sugar, it will go through the other colours first. Now, whilst that's still true, this only works with certain sugars. It works with all the sugars except sucrose. So it works with glucose, fructose, galactose, lactose, and maltose. And that is because they are called reducing sugars. Why are they called reducing sugars? They're called reducing sugars because they reduce, in other words, they cause the gain of electrons. They reduce the copper ions that are in Benedict's and make them change colour. However, it does not work with sucrose. And that is because sucrose is non-reducing, i.e. it does not reduce Benedict's. Benedict's will not change colour. So if I were to take some sucrose solution and test it with Benedict's, nothing will happen. Absolutely nothing. Now you remember we said that that sucrose, like all other sugars, is made up of two monosaccharides joined together, like all other disaccharides. So we know it doesn't work with Benedict's, but if we can break it up, i.e. hydrolyze it to form two sugars again, then we can test it with Benedict's. So how do we break up sucrose and how we test for it? So the first thing you need to do is you'll get a negative test with normal Benedict's. So we then need to hydrolyze, and I'm using the word hydrolyze rather than break because I don't like the word break in A-level biology. So I'm hydrolyze the sucrose into its monosaccharides. Now, how are we going to do that? Right, well, there are a couple of different ways you can do it, but the way we tend to do it is you're going to boil with acid. You're then going to neutralize with sodium hydrocarbonate or sodium carbonate or sodium hydroxide, whichever one you want, doesn't really matter. And once it has been boiled with acid and then neutralized, then you can add Benedict's and heat, and then it goes brick red. So the important thing is for the test for sucrose, once you've got a negative test with Benedict's, you know that no sucrose is present. You're going to need to hydrolyze the sucrose by boiling it with acid, neutralizing it, and then you can do the Benedict's test again. Okay, so now this leads on to our polysaccharides. Our polysaccharides are polymers of monosaccharides, normally glucose, joined by glycosidic bonds formed in condensation reactions. So let's start with starch. Starch is an energy storage compound found in plants. It's made up of, it's a polymer of alpha glucose. But it's made up of two types. It consists of amylose and amylopectin. And they do have a slightly different structure. So amylose is as we're used to thinking. It's a straight chain. Okay, that's important. And because it is a straight chain, it tends to form coils. So it coils up to be compact. Okay, if you're compact, that means you can store more in a small space. It only contains our 1,4 glycosidic bonds, yeah, because there's carbon number 1, carbon number 4. It's only got our 1,4 glycosidic bonds. Okay, so what about amylopectin? Well, amylopectin is a little bit more complicated. You start off with these straight chains like this. Then what you see is that you also have some branches like this. So they're attached to that carbon number 6 up there. Like that. Okay, we often just draw it like that, etc. So, important thing about amylopectin is that it's a branched chain, and because it's branched, it contains both 1,4 and 1,6 glycosidic bonds. Now, where are the 1,6, you might ask? Well, if you think about here, this is carbon number 1 up here, and there's carbon number 6 here, although I haven't drawn it. So carbon 1 and 6 have been joined together. That's our 1,6 bonds. It has many ends, and that's really important. Why are the ends important? Because if you think about what's going to happen at the ends, then what's going to happen is that when we need to release glucose, we're going to hydrolyze the ends like that to release the glucose. And the more ends that you have, the easier it is to release it, the quicker it is. Okay? Because on this chain, for example, I have 5 ends. So we describe it as having many ends so that glucose can be released quickly by hydrolysis to be used in respiration. And the other thing to know about both of these, both of them, amylose and amylopectin, is that they are insoluble. Because they are insoluble, they don't affect osmosis. So they don't draw water into the cell or cause water to leave the cell either way. So that's starch, made up of amylose and amylopectin. Amylose is straight chains, pulls up to be compact. Amylopectin is branched with lots of ends for hydrolysis of glucose, and both of them are insoluble. That's their link of their structure to their function. So what about glycogen? Well, there's not much I need to say about glycogen. And if you remember from GCSE, glycogen is the energy store in animals. If I draw it, then you will recognize it very quickly. As being very similar to amylopectin. Okay, so it's similar in structure to amylopectin. But crucially, it is more branched. Well, let's think about why that needs to be. Animals are more metabolically active. More metabolically active means you do more respiration, so therefore you need to be able to release more glucose. So it can release glucose more quickly for respiration. So it's even quicker than in plants, which aren't quite as metabolically active. Okay, and that's because there are more ends. Like starch, it's insoluble, so it doesn't affect osmosis. Fortunately, it's not quite as compact as starch is because of its branches rather than its coils. Okay, so the last one to look at is cellulose. Okay, so unlike glycogen and starch, which are both polymers of alpha glucose, cellulose is a polymer of beta glucose. Obviously still joined by condensation reactions creating glycosidic bonds, but it's a little bit more complicated than that. We know it must be joined by glycosidic bonds. We know that it's used in plant cell walls. And because it's used in plant cell walls, it must have certain features. It's insoluble, so it doesn't affect osmosis. It's got a high tensile strength. In other words, it is strong, but we're going to use more scientific words for that, high tensile strength, and it is flexible. So what we need to work out from that is how it has those functions. So we have a little bit of a problem with cellulose, because if we remember, let's draw in our molecule that we had. So here's beta, so OH is at the top and H is at the bottom on carbon number 1. So if I draw the next molecule over here, it's carbon number 4, so we've got that at the top again. Like so. We can see we have a problem. And the problem is that we don't have an OH and H next to each other. One's up there and one's down there. And we can't form a bond at a sort of diagonal. So we can't take water away, because if we take water away, then we're going to end up without our carbon-oxygen, carbon-glycosidic bonds. So what has to happen is it's going to have to flip. So what we see actually happens is that one carbon stays the correct way up, and the next carbon flips itself upside down like this. And because it flips itself upside down like so, we now have our OH at the top there and our H at the bottom there, which means that we can now remove water like we did before and create our glycosidic bonds, carbon, oxygen, carbon. So what we find is that alternate molecules are flipped. So you'll have one the right way round, like so. And then the next one will be the wrong way round, like that. And then the next one will be the right way round, like that, and so forth. Okay, so alternate molecules are flipped. So they form these really long chains. Now, if we draw these chains in, like this, just as long lines, which is what we tend to do, they all line up next to each other. And what next happens is you get hydrogen bonds. And hydrogen bonds are just an attraction between a slightly positive hydrogen, a delta positive hydrogen as we call it, and a slightly negative oxygen, a delta negative oxygen. But these hydrogen bonds, which are not that strong, but there's lots of them, they make a much stronger substance because we've got chains and chains of beta glucose molecules, which are my green lines, held together, and together they form a microfibril. So this is what provides the high tensile strength, the hydrogen bonds connecting my chains of beta glucose, forming my microfibril. And this is why plant cell walls are insoluble, have a high tensile strength, but they're still flexible.